Okay, last short little video for section 1.1. This is kind of looking at the rest of translation. So we've looked at the codons on mRNA coding for different amino acids. Now we're going to look at how that actually works and how it gets put together. Okay, so first up, we've got to make sure that we're okay with the amino acids at the end of the last little section. So the first one we already did, we said it was histidine. That might not be the easiest color for people to see. Let's try a darker green. Next one, methionine. Okay, and as I said, this is the start codon. And every single protein is going to start with that. Okay, so that's how the ribosome knows where to start actually reading the mRNA. It like searches along until it finds an AUG and it says, here, this is where I need to start. GAC is aspartic acid. UGA is arginine. GCC is alanine. And AAA is lysine. Okay, and that's just right off of that codon table. You'll always be given one. Do not need to worry about memorizing anything for it. Okay, is it possible for more than one codon to give the same amino acid? Absolutely. Okay, so 64 possible combinations of those three bases, only 20 amino acids. There's definitely more than one codon for almost all of them. AUG for methionine only has the one, and there's a couple of others. Tryptophan only has one, but yeah, for the most part they have like 2 to 4 kind of things that code for them. And the three codes for stop are UAA, UAG, and UGA. Okay, and what a stop codon means is that there isn't actually an amino acid associated with it. So when you get to one of those three stop codons, there's no amino acid joined into the chain, and so that tells the ribosome that it's done because there's nothing else for it to add on. Okay, it kind of just fits stock. Okay, so during translation, that written code, the codons for mRNA, is translated into a specific amino acid sequence by something called transfer ribonucleic acid, that's tRNA. The process always begins at AUG, which is the start codon. Okay. The tRNA is a small piece of RNA, hence RNA in tRNA, that has an amino acid that is actually attached to it. Okay. So we've got a picture of our tRNA. This top picture up here is basically showing what it actually looks like. Okay, so up at the top we've got like the twisted up, sort of back on itself piece of RNA. If you look like in here, like there's no actual bonds there. They're just like kind of beside each other, so it almost looks like a double helix, but it's not. It's just a single one that's twisted up on itself. And there's an amino acid attachment site. Okay, and that's where the amino acid actually gets attached to the tRNA, where it like pulls it out of the cytoplasm to put it there. And then you've got your anticodon at the other side. So down here, we've got our codon on our mRNA, and then we've got our anticodon on our tRNA. And the anticodon is just a set of the three bases again. RNA, so still uracil instead of thymine, and it just matches up, and that's how it knows, because if we look at like our little shapes for our bonds, like if we look at like the double versus single rings and the 2 versus the 3 hydrogen bonds, there's only like one set that can possibly match up. Okay. So that's our anticodon. So it has amino acid attached to it. So like amino acid up at the top here, so that's serine. Each of our amino acids also has a 3-letter abbreviation for it, which you don't need to know, but it's just that you don't have to write out the whole thing every time. And then your anticodon on the other side. Okay, and there's at least one type of tRNA for each of the 20 amino acids because that's how they get assembled. As the correct amino acids are brought to the ribosome by the tRNAs, they are joined together to form the protein chain that the original DNA coded for. So the DNA is like a cookbook. It's showing what you actually need there. Eventually you reach that stop codon where there is no amino acid that can be added in, and so that tells the ribosome that it's all done and it needs to stop. And the finished protein chain leaves the ribosome and moves to the Golgi body where it's packaged up and it's released so it can go do its job. Okay, so this green blob here, this is our ribosome. The mRNA is basically being pulled through it. Okay, and then you've got your tRNA coming in with, so this is your codon down here. Oh, that does not show up at all. Let's try this color. Okay, so we've got a codon here. And then an anticodon here. And then each of these is an amino acid. Okay, and as the amino acid gets joined onto the chain, we end up with a chemical bond forming between adjacent amino acids. The chain just gets longer and longer and longer. And then once it's done reading, once it hits that stop codon, then it can leave the ribosome because there isn't anything else being added onto it. Okay, so mRNA binds the ribosomal unit, which is the rRNA, the ribosomal RNA at the start codon, the AUG site. The tRNA is complementary to the site. Codon binds to the site. The next tRNA comes in, brings a specific amino acid for it, whatever is being coded for. Those amino acids bond together and form a chain. And then the empty tRNA leaves the ribosome and a new tRNA can come in. And then it continues until you hit something where there is no tRNA that matches it. Okay. And that's it. We've got some review questions. Okay. So on this whole protein synthesis thing that I would like you to work on, you've got a diagram to label. You've got a quick little summary, so what's going on kind of at each step. So what is happening at step 1 all the way through to step 9? What's going on there? Okay. And then that's kind of our overview of what this actually ends up looking like. So we've got our DNA, and then a small little section of the DNA is a gene. Okay, so there's our gene. And then here's another gene, and then here's another gene. Okay, so different sections of the DNA have information for different genes on it. It gets unzipped so you can see the DNA strand, the singleton one. It acts as a template for your mRNA to basically match up to it. And you can see kind of all of these shapes line up really nicely. And then your groups of 3 for your mRNA are considered to be a codon. And then translation occurs where your tRNA brings in the amino acids that come together and form your protein. So that's kind of your overview of the whole thing. Okay, and that's it for section 1.1. So then we've got DNA and protein synthesis review. So lots of review questions for you to work on. And on Friday's live session, we'll go through kind of all the answers to these. Okay.